sexual reproduction in flowering plants, right? So already we saw in detail about the flowering plants. So what are flowering plants? That is the angiosperms. We studied even in 11th standard about the different types of plants like algae, bryophytes, pteridophytes, gymnosperms and angiosperms, right? So, and also we saw everything about the angiosperms, about the structure of the flower. So today we'll see in brief about the structure of the flower and about the pre-fertilization. So what do you mean by pre-fertilization? That is, what are the things which are required for fertilization? That is for the fusion of male and female gametes, right? So here under the pre-fertilization, particularly in flowering plants, so we'll see about the male and the female reproductive structure, that is andrecium and gynoecium. So first there must be the development of the male and female reproductive structures, right? And we'll see in detail about stamens, microsporangium and pollen grains. So what are stamens? Stamens are the male reproductive structures and how the pollen grains are formed. So we'll see in detail about the structure of the flower, pre-fertilization structure and events, and about the andrecium, that is stamen, microsporangium and pollen grains. So, okay. So what is sexual reproduction? Already we saw that there occurs a difference between the asexual reproduction and the sexual reproduction. So sexual reproduction, it involves the two partners, right? And also it involves the formation of male and female gametes. So all the flowering plants, whatever plants we see around, most of the plants, they have the colorful flowers, right? So what are the role of flowers? So flowers are the main agents that show sexual reproduction, right? So these flowering plants, they show sexual reproduction, and flowers are the sites of sexual reproduction. So they have the male and the female reproductive structure. So we already saw in detail about the structure of the flower, right? So there are 4 whorls. One is calyx, that is corolla, that is sepals, then petals, then the male reproductive structures are andrecium and gynoecium, right? So these two are the male and the female reproductive structures, and they are mainly involved in fertilization, right? So we know the structure of the flower, that is the sepal. So this is the sepal. Then petals, the colorful petals are present. The main purpose of the colorful petals are to attract the insects and other animals, and they aid in pollination. And this is the male reproductive structure, that is stamen, which consists of an anther and a filament. This stalk-like structure is the filament. And this is the female reproductive structure, right? They have the stigma. So the pollens will come and stick to the stigma, and they lead to the formation of the pollen tube inside. So here there will be the formation of pollen tube, right? So through the pollen tube, the pollen will enter the ovary, and here they fuses with the egg and leads to the fertilization. So this is the general structure of the flower. Actually, we already saw in detail about the structure of the flower. So you are clear with these structures, right? So we'll now proceed to the pre-fertilization structures, right? So you are clear with the different whorls. So if all the 4 whorls are present, the flower is called the complete flower. And if any one of the whorl is not present, it is called an incomplete flower. And also we saw in detail about the unisexual and the bisexual flowers, right? So in addition to that, there are different terms like pedicel. So what is a pedicel? Thalamus. So the flower has a stalk which is called the pedicel, so pedicellate flower, and then thalamus is the enlarged part which is present at the base, and they have the bracts, that is leaf-like appendages, which are present at the base. And if the plant, they have the bract, it is called bracteate, and if it doesn't have bract, it is called the ebracteates. So I hope you are clear with all these terms, right? So we already saw in angiosperms. So today we'll see about the pre-fertilization structure and events, and in detail about androecium. Okay. So several hormonal and structural changes that result in the differentiation and development of floral primordium. So primordium is nothing but the earliest stage of the development. So these hormonal and structural changes that is happening in the flowers, it leads to the development of the flower. So this actually we saw already about the inflorescence, right? So the inflorescence, they bear the floral buds and then the flower. So then it develops the male and the female reproductive structure. So what are the changes that are happening due to the hormonal changes and the structural changes? It results in the development of the pre-fertilization structures, that is the formation of androecium and the gynoecium. Okay, so we'll see in detail about the androecium and gynoecium. So pre-fertilization structures and events, it involves the androecium and gynoecium. So which one is androecium? Androecium is the male reproductive part. So this is the androecium. So if you see the androecium, it has the filament and the anther. So this is the anther. So it is a stamen. Stamen, it is also called as microsporophyll. Stamens also called the microsporophyll. Okay, so they develop into microsporangia. So we'll see in detail about the microsporangium and how the pollen grains are formed. Okay, so this is the male reproductive structure. It is a stamen which consists of filament and the anther. Okay, and gynoecium is the female reproductive part, that is carpel, and it has a stigma, style, and the ovary, which has the ovules, which is also called the megasporangia. Okay, so if you take the androecium, it is the male reproductive part of the flower. So androecium is the male reproductive part of the flower, and it consists of whorl of stamens. That is, many stamens are present, but their number actually differs among different species, right? So the number of stamens and the length of stamen, it varies among different species. So the stamen consists of filament and the anther, right? It has the filament and the anther, and they are connected by the tissue that is called the connective tissue. So all this structure we saw already, that is pedicel, stalk of the flower, and this is the floral axis, and petals, that is they form an accessory whorls, right? Corolla and calyx, they form the accessory whorls, that is sepals and petals. In some cases, the sepals and petals are not differentiated and they will be together, and they are called the tepals, right? And the whorl is called the periantha. And this is the ovary which has the ovule. So this is the structure of a mature flower. And if you take the filament, so what is the role of filament? It is a long and a slender stalk. So it is a long structure and which carries the anther, right? Its proximal end is attached to the thalamus or the petal of the flower. So thalamus is present at the lower part, right? So this filament is attached to the thalamus or the petal of the flower. And then at the tip of the filament is the anther. So it is present at the terminal end, and it is typically bilobed. That is, it is present in the form of two lobes, right? It is present in the form of two lobes, and each lobe has two thecae. So each lobe will have the two thecae, and we are called it as dithecous condition. So dithecous, and if you see the structure again, so this is the structure, this is the filament, and this is the anther. Here it is two lobed, so it is called dithecous, right? And often a longitudinal groove, it runs lengthwise, separating the theca. So in the in between we can see the separation, right? So this is a longitudinal groove that runs between the two theca. And if you see the transverse section of the anther, so it is a tetragonal structure. So this anther, it is a tetragonal structure and it consists of 4 microsporangia. So we can call it as tetrasporangiate. Tetrasporangiate. Okay, so it consists of 4 microsporangia, which is located at the corner. So in each lobe, there will be the 2 microsporangia, right? So there are 4 microsporangia. And this microsporangia, it develop into pollen sacs. So if you see the tip, see here, these are the pollen sacs. So in between, there is a separation, and this microsporangia, it develop into a pollen sac, and this pollen sac is packed with pollen grains, right? So this is a filament, and this is the top part, that is the anther, and it contains the microsporangia, and this microsporangia develop into pollen sacs. Okay, so the microsporangia forms into a pollen sac, and they extend longitudinally all through the length of the anther. So they are present throughout the anther, and they are packed with pollen grains, right? So this pollen sac is filled with pollen grains. So if you take the structure of microsporangium, so where the microsporangium is located, that's what we saw, the anther, that is in the upper part, right? The anther, it consists of 4 microsporangia. So now we are going to see in detail about the structure of microsporangium, right? So the typical microsporangium is near circular in outline. So it is circular in shape, and it is surrounded by 4 layers. That is, the outer one is epidermis. So this is the structure of a microsporangium, right? The microsporangium, where is it? It is present in the anther. So here it is surrounded by 4 layers, epidermis and endothecium. Innermost is the, that is innermost to epidermis is the endothecium, and then there are middle layers, and then the innermost is the tapetum. So these are the 4 layers that are present in the microsporangium. Okay, it is surrounded by 4 wall layers, and among these, the 3 outer layers. So which are the 3 outer layers? It is epidermis, endothecium, and the middle layer. The purpose of this is protection, right? So they give protection, and they help in the dehiscence of anther to release the pollen. So what should happen? The pollen grains are present in the pollen sac, so they have to dehisce. So this microsporangium layer, it has to dehisce to release the pollen, right? So these 3 outer layers, they give protection, and they help particularly the endothecium, which is involved in the dehiscence to release the pollen. So these 3 outer layers are responsible for protection. Okay, so what is the role of tapetum? So tapetum is the innermost layer, and it nourishes the developing pollen grains. So it gives nutrition, right? The outer 3 layers, that is epidermis, endothecium, and the middle layer, they give protection. But the innermost layer, tapetum, it provides nutrition to the developing pollen grain. Okay, so the cells of the tapetum, it contain dense cytoplasm. So they have more than 1 nucleus. So we'll see in detail about the type of the tapetum that is present. Okay, so they contain generally more than 1 nucleus. So if you take an young anther, each microsporangium has a sporogenous tissue. So this microsporangium, they have the sporogenous tissue at the center, and it consists of compactly arranged cells, which are called sporogenous cells. So these sporogenous cells, they actually acts as a pollen mother cells. Okay, so we saw about the 4 layers, and the outer 3 layers are involved in protection, and the inner layer gives nutrition, right? So the cells of the tapetum, they have the dense cytoplasm, and this microsporangium, they have the sporogenous tissue and consists of sporogenous cells. Okay, so if you take the tapetum, there are two types of tapetum. The first type is amoeboid tapetum. Amoeboid tapetum, which is also called the invasive or periplasmodial tapetum, or invasive or periplasmodial tapetum. So this is actually found in primitive angiosperms. So it is a primitive form, right? It is found in primitive angiosperms, and it absorbs, the main role of it is it absorbs food from the middle layer. Absorbs food from the middle layer. And it provides nutrition to the developing microspores after degeneration. Actually, this is a primitive type and it degenerates quickly. But there is another type of tapetum which is called the glandular tapetum or secretory. So this is an advanced type of tapetum and it does not degenerate quickly. Does not degenerate. So this is usually found in most of the flowering plants, glandular or secretory tapetum that is found. And there is another term that is called the pollen kit. So what do you mean by pollen kit? For example, in the case of plants in which the pollination occurs by means of insects, there are various pollinating agents like insects, animals, right? So those plants which are pollinated by insects, so those pollen grains, they have an oily layer. The pollen grains will have an oily layer around the pollen, right? So this oily layer is particularly due to the presence of the carotenoids and other lipids. So they form an oily layer. So what is the purpose of this oily layer? So that they can be easily, that is transported by means of the insects. It will stick to the insects, right? So they have an oily layer. So that is called the pollen kit. So this function of this tapetum is actually, it helps, it provides nutrition. One thing is it provides nutrition, right? Provides nutrition to the pollen mother cell. And the second function of this tapetum is secretion of sporopollenin. Pollenin and also pollen kit in some cases. Pollen kit materials. And third main function is secretion of enzymes and hormones. Enzymes and hormones. Okay. And fourth main function is it is involved in the formation of substances that is called the Yubish bodies. So what are these Yubish bodies? So actually this cells of tapetum and the middle layer, actually they will degenerate. But before degeneration, this they form the granules first. They call the pro-Yubish granules or the pro-Yubish bodies, or which is also called the Yubish bodies or orbicules. So this pro-Yubish bodies, they are surrounded by the sporopollenins. So that is called the Yubish bodies. So the role of this Yubish bodies is that when the tapetum degenerates, that there occurs a degeneration of tapetum, right? So when it degenerates, this Yubish bodies, they are released into the pollen sacs. they are released into the pollen sacs and they are involved in the formation of exine layer. So what is the exine layer? It is the outermost layer of the pollen grain, right? So this ubish bodies are involved in the formation of exine layer. Okay, so the main functions of the tapetum are it provides nutrition to the pollen mother cell and secretion of sporopollenin and pollen-capped materials, and it is also involved in the secretion of enzymes and hormones, and it is involved in the formation of ubish bodies or orbicles. Okay, so the role of this ubish bodies is they are involved in the formation of exine of pollen grains. Okay, so are you clear with this? So now what happens when the anther develops? That is the development of anther. So when it develops, each sporogenous cell, so the cells which are present, they are the diploid cells and they are called the pollen mother cells. That is each sporogenous cell, it undergoes meiotic division to form microspore tetrads. So they form the tetrads. That is this is the structure of a microspore tetrads, right? So each sporogenous cell undergoes meiosis. So this is the mother cell, or we can say pollen mother cell, which undergoes meiotic division to form microspore tetrads, right? So each cell, they form 4 tet cells. The formation of microspores from a pollen mother cell, this process is called microsporogenesis. That is the formation of microspores. So genesis is the formation. So there is a formation of microspores, right? So the tapetum is the innermost layer, and here the micropollen mother cells are present, and this pollen mother cells, they are diploid cells, so they undergo meiosis and they form tetrad of pollen. That is pollen grains are formed, right? So this is the structure of microsporangium. So as the anther mature, so when they develop and they mature and dehydrate, the microspores dissociate from each other. So we saw that they are produced in the form of tetrad, right? So these tetrad, actually they are associated with each other and they are degraded by the enzyme calase. And this enzyme is present in tapetum. So tapetum, actually we saw that they secrete hormones and enzymes, right? So the calase enzyme, it causes the degradation of this microspore tetrads into the microspores. So they dissociate from each other and they develop into pollen grains, right? So first they are formed in the form of tetrads, then they dissociate and develop into pollen grains. So this shows the dehiscence of anther, mature dehisced anther. So here they release the pollen grains. Okay. So each microsporangium consists of thousands of pollen grains. Thousands of pollen grains are present in each microsporangium and they are released with the dehiscence of anther. So they liberate, they burst out and they release the pollen grains, right? So, and now we'll see in there. So are you clear with the structure of microsporangium and any doubts on that? Clear, right? Okay, so now we'll see about the structure of pollen grain. So now the pollen grain is released, right? So the pollen grain is generally, it is spherical in shape. So the pollen grains are generally spherical and the diameter is about 25 to 50 micrometer in diameter. And the cytoplasm is surrounded by a plasma membrane. So the pollen grain is surrounded by a plasma membrane and it has a 2-layered wall, exine and the intine. So the outer layer is the exine and the inner one is intine. So we'll see about the structure of the pollen grain. So this exine is the hard outer layer and it is made up of sporopollenin. So sporopollenin is the hardest substance. So it is highly resistant For example, for strong acids or the alkali treatment, they are highly resistant. So it is composed of sporopollenin and it is a highly resistant organic material. So the sporopollenin is, so as a result of it, it can withstand the high temperature and acid and the alkali treatment. And also another special feature about this is the enzymes cannot degrade sporopollenin. So it is able to withstand all sorts of conditions because of the presence of sporopollenin. Okay, so it has an apertures which are called germ pores. So it has the pores. In some areas there will be no sporopollenin. So if you see the structure of a pollen grain, there will be some areas which are called the germ pores. So this is the germ pores, right? And some places the sporopollenin is not absent, so that is called the germ pore. So based on that, there are different types, like based on the number of germ pores. For example, if 3 germ pores are present, then the condition is called tricalpate. This is particularly present in dicots, and when only 1 germ pore is present, it is called monocolpate. And this type of pollen is particularly present in monocots. Right? So based on the number of germ pores. So hexine has an aperture called germ pores where sporopollenin is absent. And pollen grains are preserved as fossils due to the presence of sporopollenin because of its highly resistant nature. So as it is a highly resistant organic material, so they are preserved as fossils, that is, they will not degrade so easily, right? So they exhibit patterns and designs. Hexine. So hexine is an outer layer of a pollen grain. And the inner layer is called the intine. It is the inner wall. And it is a thin layer and it is a continuous layer which is made up of cellulose and pectin. So it is a thin layer which is present inside. And a mature pollen grain, if you see the structure of a mature pollen grain, it consists of 2 cells. One is called a vegetative cell, the other one is called a generative cell. So the vegetative cell is generally bigger and it has abundant food resources. So it helps in the storage of food material and has an irregularly shaped nucleus. So that is a vegetative cell. So this is how the microsporangium, it is converted into a pollen grain, right? So the mature pollen grain will have the 2 cells. One is a vegetative cell, the other one is a generative cell. So the vegetative cell is usually bigger and it has abundant food reserve and has irregularly shaped nucleus. Okay, and generative cell is smaller in size and it floats. So this is the generative cell and this is the vegetative cell. So here it is smaller and it floats in the cytoplasm of the vegetative cell and it is spindle shaped with dense cytoplasm and a nucleus. So if you see the many of the angiospermic plants, we know that there occurs the shedding of pollen grains. So usually at this stage, that is the 2 cell stage, one is a vegetative cell and another one is with the generative cell. They shed their pollens in 60% of the angiospermic plants. Okay, they shed their pollen at 2 cell stage. But in others, what happens? This generative cell, that is a small cell which divides again mitotically. So again there occurs in mitotic division to give rise to 2 male gametes. So here again 2 male gametes are formed. So the pollen grains are shed at the 3 cell stage. So in the case of angiospermic plants, the pollen grains may be shed either in the 2 celled or in the 3 celled stage. Okay, so the pollens may be shed either in 2 of these stages. So now if the pollen grains are shed, so what is the next step? The pollen grains have to land on the stigma. right? So now the pollen grains are formed and the pollen grains have to land on the stigma for an effective fertilization to occur. So the shed pollen grains have to land on the stigma before they lose their viability, because they will be viable only for a shorter period. That too, it differs among plants. So they have to land on the stigma before they lose viability, right? So the viability period of the pollen grains is variable. So it also depends on temperature and the humidity. But in some cases, like rice, wheat, that some cereals, only 30 minutes. That is, the pollen grains are viable only for 30 minutes. So within that period, they have to land on the stigma so that effective fertilization can occur. But some members of Leguminaceae, Rosaceae, and Solanaceae, they have the viability for months. So they are viable for a longer period. Okay, so it differs from plant to plant. And if you see the economic importance of the pollen grains, so they are rich in nutrients. So these pollen grains are rich in nutrients. So the pollen tablets, so as they are rich in nutrients, so they are nowadays using it as a food supplement for giving the nutrients, right? So they are used as food supplements, and this pollen tablets and syrups, they increase the performance of athletes and racehorses. So if you give the pollen tablets, as they are rich in nutrients, their performance rate, that is, give more energy, right? So they increase the performance of athletes. And also they are stored for years in liquid nitrogen at minus 96 degrees centigrade. So what is the main purpose of it? So we can store the pollens and we can use for the crop breeding or the artificial breeding programs, right? So they are used as pollen banks. They store the pollens of different plants. The pollen grains of some plants, example, Parthenium or carrot grass. So Parthenium, you know that many people are allergic to it, right? So the pollen grains are particularly responsible for this allergic reactions and leads to chronic respiratory disorders like asthma and bronchitis. So the pollen grains are allergic to some people, right? So I hope you are clear with this, right? Any doubts on this till now? So today we saw in detail about the stamens and also about the microsporangium and about the pollen grains, right? So you're clear with it. So how the cells are formed and how these pollen grains. Now in tomorrow's class we'll see in detail about the megasporangium. So next step is the how this pollen enters into the, that is how it lands on the stigma and how it leads to the formation of the pollen tube and how it reaches the ovule and how the fusion takes place. Okay, so clear? So microsporangium is generally surrounded by 4 layers. Which of the following 3 layers perform the function of protection and helps in the dehiscence of anther to release the pollen? So what is the correct answer for this? Epidermis, tapetum and endothecium, epidermis, aril and endothecium, epidermis, endodermis and mesocarp, epidermis, middle layer and endothecium. Yes, D is the right answer because these 3 layers are the outer layers, right? Epidermis, middle layer and endothecium. So these 3 are the outer 3 layers which are involved in protection, but tapetum is the inner layer which gives nutrition. So each cell of sporogenous tissue in anther is a microspore, pollen, potential pollen or microspore mother cell and megaspore mother cell. So the question is, each cell of sporogenous tissue in anther is, each cell of sporogenous tissue in anther is whether it is a microspore, pollen or the potential pollen or microspore mother cell or megaspore mother cell. Yes, C is the right answer because it is a pollen mother cell or a microspore mother cell, and this pollen mother cell or the microspore mother cell. So what will happen? They undergo meiosis and they form the microspore tetrad, right? So each cell of sporogenous tissue in anther is a potential pollen or microspore mother cell. Pollen grains can be stored in liquid nitrogen at 70 degrees centigrade, 100 degrees centigrade, minus 196 degrees centigrade, or 0 degrees centigrade. Yeah, C is the right answer because it is stored at minus 196 degrees centigrade so that it can be used for the crop breeding programs. So which of the following points is incorrect about sporopollen? It is one of the most resistant organic material known. It can withstand high temperature and strong acids and alkali. Two enzymes that degrade sporopollen are known so far. Pollen grains are well preserved at fossils because of the presence of sporopollen. Yes, C is the right answer again here. So why C is the right answer? Because that is the incorrect statement. No enzyme actually that degrade the sporopollen are known so far, but here it is given as two enzymes. So all the other statements are correct. It is the more resistant organic material known, and it can withstand the high temperature and strong acids and alkali, and pollen grains are well preserved as fossils because of the presence of the highly resistant capacity of the sporopollen. So C is the right answer. The proximal part of the filament of stamen is attached to thalamus or the petal, sepals or the thalamus, pedicel or petiole, ovary or ovule. So which is the correct answer? The proximal part of the filament of stamen is attached to which part? Yes, A is the right answer. That is thalamus or the petal. Okay. So A is the right answer. So we'll discuss few questions. So the first question, the science of cultivation, breeding, then and arrangement of the flower is called arboriculture, floriculture, horticulture, and anthology. So what will be the correct answer here? The science of cultivation, breeding, and arrangement of the flowers is called the floriculture actually. Floriculture is the science of breeding, and arrangement of flowers. Okay, it is called the floriculture. The non-essential floral organs in a flower are sepals and petals, anther and ovary, stigma and filament, petals only. So what is the correct option? The non-essential is A is the right answer. That is sepals and petals. Non-essential floral organs in a flower are sepals and petals. So they are called the accessory whorls or non-essential because what is the role of sepals and petals? Only that is they are not involved in the fertilization. The antecium and gynoecium are involved in fertilization. So these are called the non-essential or the accessory whorls, right, or floral organs. So A is the right answer. Then the next question. Anther is generally monosporangiate, bisporangiate, tetrasporangiate, or trisporangiate. Anther is generally monosporangiate, bisporangiate, tetrasporangiate, and trisporangiate. Yeah, they are tetrasporangiate, right? Then the anther wall consists of 4 wall layers, where option E, tapetum lies just inner to endothecium. Middle layers lie between endothecium and tapetum. Endothecium actually lies inner to middle layers. Tapetum lies next to epidermis. So which is the right option here? Yes, B is the right option, that is the middle layers lie between endothelium and tapetum. So you have to be clear with the order. It is epidermis followed by endothelium, and which is followed by the middle layers and tapetum. So middle layers lies between endothelium and tapetum. And callase enzyme, which dissolves the callose of pollen tetrads to 4 separate pollens, is provided by pollens, tapetum, middle layers, and endothelium. So what is the correct option here? That is the callase enzyme. So which one secretes the callase enzyme? Which dissolves the callose of pollen tetrads to 4 separate pollens is provided by pollens, tapetum, middle layers, and endothelium. So are you able to follow this question? Actually, we saw, yeah, B is the right answer. Because we saw that the microspores are formed in the form of tetrads, right? So here the tetrads, they are connected by means of the callose. And this callose is degraded by the enzyme callase. And this callase enzyme is secreted by tapetum. Okay, this is secreted by tapetum. So only tapetum, we saw the functions of tapetum, right? It is involved in the secretion of enzymes and hormones. So this callase is the main enzyme which is secreted by the tapetum, and it helps in the degradation of the callose which joins the microspore tetrads. Okay, clear? The next question, which function of tapetum is correct? It helps in pollen fall formation and helps in the transportation of nutrients to inner side of the anther, synthesis of callase enzyme for separation of microspore tetrads, all of these. So what is the correct option? Yes, D. All of these are functions of tapetum. So it helps in the pollen wall formation. Pollen wall formation is, it is the 4th layer, right? So it helps in the pollen wall formation, and it helps in the transportation of nutrients. So the main role is to provide nutrients to inner side of the anther, and it is also involved in the synthesis of callase enzyme for the separation of microspore tetrads. Then the next question is, in angiosperms, various stages of reductional division can be best studied in young anthers, mature anthers, young ovules, and endosperm cells. In angiosperms, various stages of reductional division can best be studied in which one? Whether it is an young anther, mature anther, young ovules, or endosperm cells. Yes, A, that is young anthers. We can study the various stages of reduction division, that is meiosis, and how the microspore tetrads are formed, right? So the study of pollen grains is called, so it is called microbiology, anthology, palynology, or pomology. So the correct term is palynology. Yes, C is the right answer. This is the study of pollen grains. Right? It's called palynology. So how many pollen mother cells should undergo meiotic division to produce 64 pollen grains? So if 64 pollen grains are to be formed, how many pollen mother cells should undergo meiosis? So the options are 64, 32, 16, and 8. Yeah, 16 is the right answer. So I hope you're clear with the calculation, right? So each pollen mother cell, it will produce 4, that is 4 microspore tetrads we know. So 4 pollen grains. So if 64 pollen grains are to be formed, 16 pollen mother cells should undergo meiotic division. Yeah, 16 is the right answer. That is option C. And how many meiotic divisions are required for the formation of 100 pollen grains? So if 100 pollen grains are to be formed, how many meiotic divisions should happen? That is 100 or 50, 25 or 26. So what is the correct option? Yes, 25C is the correct option, right? So 25 meiotic divisions are required for the formation of 100 pollen grains. Okay. Then what is the function of germ pore? It is the emergence of radical, absorption of water for seed germination, initiation of a pollen tube, and all of these. So it is the correct option here. The function of germ pore: emergence of radicals, absorption of water for seed germination, or initiation of the pollen tube, and all of this. So here the correct option is initiation of pollen tube. So what happens here is initiation of pollen tube. C is the correct answer. So the germ pore, what is the role of germ pore? The germ pore is a place, that is in the case of pollen grain, what is the structure? This is the exine and inner one is the intine, right? And here it will be exine. And here in the exine, there are some places where no sporopollenin or sporopollenin is absent, right? So that area is called the germ pore. So what happens is this exine, intine, actually it bursts out when it, for example, this is the pollen grain, it lands on the stigma. Pollen grain, it lands on the stigma, right? So when it lands on the stigma, what happens is this will take up the nutrients from the stigma. So here it leads to the formation of the pollen tube. So how the pollen tube is formed is through this germ pore. The intine will come out through this and they form the pollen tube. So function of the germ pore is to initiate the formation of the pollen tube so that it can, so other places there will be a sporopollenin. So through this germ pore, the intine comes out. So it helps in the formation of the pollen tube. Okay, so that is the main function of germ pore. So C is the right answer. So you're clear with that, right? So the next question is, one of the most resistant biological material present in the exine of pollen grain is pentocellulose, poropollenin, suberin, and cellulose. So it is the correct option. Yeah, B, sporopollenin. And which of the following statements about sporopollenin is incorrect? Exine is made up of sporopollenin, and sporopollenin is one of the resistant organic material. Exine has apertures called germ pores, where sporopollenin is present, and sporopollenin can withstand high temperature and strong acids. So which is the right answer? So which of the statement here they asked is incorrect? Which one is incorrect among this? Yeah, C is the incorrect statement. So why C is incorrect? Because it is given, it has apertures called germ pores where sporopollenin is present. But in the pores, sporopollenin is absent. So C is the right answer. Okay. Dash of the pollen grain divides to form two male gametes, whether it is a vegetative cell, generative cell, microspore, mother cell, or none of these. Dash of the pollen grain divides to form two male gametes. Yes, B, only the generative cell, right? This generative cell, it divides to form two male gametes. So we saw that vegetative cell and generative cells, right? And the pollen grain may degrade or it may shed at the 2-cell stage or it may shed at the 3-cell stage. So this divides to form two male gametes. And cells that are found in a pollen grain when it is shed at 3-cell stage, or whether it is an one vegetative cell, one generative cell, and one male gamete. That is, the options are whether it is one vegetative cell and one generative cell, and one male gamete. or it is one vegetative cell and two male gametes, or it is one generative cell and two male gametes, and either A or C. So what is the correct option? No, the correct option is B actually. So it divides the cells that found in the pollen grain when it is shed at a 3-cell stage. So at the 3-cell stage, what will happen? There will be a vegetative cell and the generative cell is actually divided into two male gametes, right? So B is the right answer here. One vegetative cell and two male gametes. Okay, clear? So the next question. So in a fully developed male gametophyte, that is a pollen grain, it forms a male gametophyte. So in a fully developed male gametophyte of flowering plants, the number of nuclei is 1, 5, 3 or 4. In a fully developed male gametophyte of flowering plants, the number of nuclei is 1, 5, 3 or 4. So the correct option is C. That is, they will have the 3. In a fully developed male gametophyte, that is, they will have the 3 nuclei. One in the case of a vegetative cell and other 2 male gametes are formed. So they will have the 3 nuclei. So C is the right answer. Clear? So if you go to the, that is what is happening when there is a formation of the pollen grain. We saw, right? Pollen grain is the initial stage and it develops into a male gametophyte. Pollen grain, it develops into a male gametophyte. So first, as a result of cell division, it forms vegetative cell and a generative cell. Right? And this generative cells, it again divides and it forms the 2 male gametes. So it may be either shed in the 2-cell stage or in the 3-cell stage, right? So in the 3-cell stage means actually they will have the 3 nuclei, right? Are you clear with this? It may be either shed at the 2-cell stage. In 60% of the angiosperms, they are shed at the 2-cell stage, or it may be shed at the 3-cell stage. So when the 3-cell stage is happening, what is happening, how it is becoming the 3-cell? That is, it is split into 2 male gametes. Okay. So when the pollen, it lands on the stigma, what happens is, through the exine, it has a sporopollen, and so it cannot come out. So this pollen lands on the stigma. It uses the nutrients from the stigma, and this exine, through the germ pore, the intine comes out of it. First, the vegetative cell, there occurs the formation of the, it initiates the formation of pollen tube. So through the pollen tube, which went first the vegetative cell, it enters into the pollen tube, which is followed by the 2 male gametes. So how the pollen tube is formed? Through the germ pore. So the main function of the germ pore is the initiation of the pollen tube. So after it lands on the stigma, it initiates the formation of the pollen tube. So first the vegetative cell enters, and then the 2 male gametes, it enters into the ovary. Okay, so this is the general steps that are involved in the formation of the pollen tube. So you're clear with this, right? So tomorrow we'll see in detail about the structure of gynoecium, or the female reproductive part. So these two, that is androecium and gynoecium, that comes under the pre-fertilization structure and events, right? So after the gynoecium and the androecium is formed, then there occurs an fusion or the fertilization. So that is brought about by various pollinating agents. So next we will see about the types of pollination and about the various types of pollinating agents. Okay, so clear, right? So any doubts? Okay, so we'll continue tomorrow then about the gynoecium. Okay, so you go through it and read all these steps and mainly about these processes. So how the pollen, first step is stamens, anther, filament, and in the anther, so how it leads to the formation of microsporangium and then to pollen grains. So from the pollen grain, it forms the male gametophyte, right? So just you read through the steps and go through the diagrams clearly. So you read again and again, right? So NCERT book is more important here. So you go through all the diagrams. Okay, so we'll continue tomorrow. Thank you.